Okay, well, it's been a fantastic afternoon. I've learned a lot. I think that the team that's been assembled here has been quite revealing to me, because I know a lot of the people that have been involved with setting up this wonderful program here. But I think there's some tremendous opportunities moving forward. So what I'm going to do is, most of the people that gave talks sent me a couple of slides. And so what I've tried to do is to bring this together and perhaps make some commentaries on not just what I think the opportunities are as have been identified by the speakers so far, but also where the total body PET facility here exists within the constellation of research in PET within Australia and also internationally, and perhaps some ideas that have come to my mind even during the talks today as to where the opportunities are moving forward. Now, of course, you know, it's hard to imagine someone with more background, skill, knowledge, and we're very privileged to have Simon, who's been here to be able to present the history of PET in general, but also total body PET. And I was fascinated to hear about how many grants he had to apply for before he was actually able to get this up and running. And for those of us who submit a lot of grants, we can sympathise, because good ideas don't necessarily get funded first time. In fact, usually they don't. And so we heard all about the story of how the first total body instrument was created, the scans that were started, the possibility of doing dynamic imaging. And we've heard throughout the afternoon a number of speakers talking about how important it will be and the opportunities for being able to do dynamic imaging. And I think Steve, in his commentary about what would George do, I knew that was the title of his talk. I wasn't quite sure what he was going to talk about. And I think there was it posterior density analysis or something? I've no idea what that means, but it sounds cool. So I'll have to ask you a bit more about it later on. And that obviously links to the kinetic modelling and the opportunities for looking at total body and organ interactions. For those of us who've been in nuclear medicine for a while, we have been looking at dynamic imaging. You know, in the cardiac space, looking at first pass studies is something that we've all been doing for many years, but probably reducing in number over the last, say, 10 years. So opportunities moving forward clearly will be huge. The images that have been shown from a clinical perspective are of course transformative. You know, the ability to be able to see lesions in much more detail in a fraction of the time is going to be incredibly important. And in fact, a comment was made, well, how are you going to be able to image or interpret the images now? It's a bit like every time a new technology advance has occurred with PET, going from the original older crystal PET scanners through to the newer generation, PET-CT, now total body. We have to adjust what's normal versus what's abnormal, and that's part of the learning process with all of these. But clearly the ability to do fast scans, to be able to reduce the dose is incredibly important purely just from a clinical perspective. And I was very interested in the presentation from Kevin London, who talked about pediatrics. And of course, that's an area where there's an immediate utility for the use of total body PET. And I was very impressed with the protocol which has been proposed to do the comparison between static imaging and then single day and the total body PET instrumentation scans that will be done. And the differences between, you know, the classic MIBG scans, which we all grew up with and which we've performed, through to the new generation MFBG and total body systems, I think will be transformative for them as well. Now, tracer kinetics are clearly, as we've just heard in a very elegant talk by Steve, important. And I should say Geiger-Müller counters are still useful. We use them almost every day for our inpatients who have been treated with radioiodine, or for our patients that are getting some of our other therapeutic isotopes, because we need to know just how much activity there is and if they can in fact leave the department or the inpatient rooms and be discharged from hospital. But of course, being able to look at kinetics is totally different when you can do total body dynamic scanning. And I do think that that is an area where this facility will be able to make a huge contribution in many different ways. And I'll talk about that over the next few slides. We heard about how there's this incredible linkage between what's happening in the brain and other organs in the body. Often when we do total body scans, as part of routine clinical scans, we image the brain and we have a cursory look at it, but we don't necessarily look at it in great detail unless there's some obvious neurodegenerative changes present. But the opportunity to be able to more interrogate both the brain FDG uptake as well as the rest of the body in selected patients where you do dynamic imaging could in fact very quickly result in cohort sizes that would be able to address some of the key questions that have been raised as part of the presentation and discussion here today. We don't know what those linkages are. That could be done with the total body scanner. And of course, we've just heard also about the linkage between heat stroke, gut permeability, cardiovascular reserve. I was mentioning to Paul and to Dale just at one of the breaks that one of the things that you can actually do with just a standard PET CT scan, and we've just completed a small cohort at our institution, is you can actually do coronary artery calcium scores from the actual attenuation correction CT scan. It is possible. And if that could be combined with some of these other dynamic studies, it might be that that could also contribute to looking at patients that are at higher risk of being in the context of heat exposure, more likely to actually develop heat stroke. But these are things which, from a population basis, I think could easily be looked at, even in the clinical cohorts that you're studying here as well. I've got a feeling of a grant coming on there as a way of being able to actually test this at multiple sites. And I think that's an important point, is that through NHMRC, and particularly the Medical Research Future Fund, there is quite good funding now for multi-centre trials, of which PET, as I'll discuss later, has been quite successful over the last 4 or 5 years. 5 minutes, okay. So we heard from Dale about the ability to use total body PET for clinical translation. And I think that that's clearly an area where really it's just a matter of what our vision and our ideas can really generate. There is almost no limit to the number of targets and body functions for which you can't develop a PET tracer to interrogate. And if there's one thing that I would like to mention, which hasn't been touched on perhaps a lot during this afternoon session, is that having innovative chemistry is critical to achieving the outcomes from this total body facility, which you can generate. Just a few examples that I'd throw in here is that in the cancer space, immunotherapy has revolutionised treatment, but we still don't know which patients respond best. Imaging of the actual target, such as in the case of PD-L1 imaging with pembrolizumab or durvalumab, is one way in which you can actually identify patients that may in fact better respond. But most of these studies have been done with relative static imaging as opposed to dynamic imaging. Wouldn't it be wonderful to be able to do that? Through the National Imaging Facility, there is in fact a molecular imaging and radiochemistry network which can link sites around the country. So new probes can be generated at one site and transferred to another in a fairly seamless way. An example of this is a PD-L1 imaging probe durvalumab, for which we've developed an automated synthesis technique, which is now being used at 3 sites around the country. Could easily be done here at North Shore Hospital as well. And there are many other examples of that as well. And this is just an example where we've applied this in the context of lymphoma patients and been able to show a direct correlation between the uptake of the PD-L1 durvalumab and the actual subsequent response in this particular patient cohort. And these are, difficult to image studies because they're with a zirconium labeled antibody. And so these images, you don't want to know how long they take to actually perform, but you could do them here in 5 minutes. It would be absolutely outstanding. There are, in fact, 4 clinical trials that are being conducted around the country at the moment, investigator-initiated, with the zirconium-labelled durvalumab. And so that's just an example of one probe which could also be evaluated here. And the imaging that you could do would be outstanding and really complement what's happening elsewhere. We heard also about the imaging of CD8 T cells. Simon might be interested to know there are actually 5 clinical trials with this probe happening in Australia at the moment, where this particular probe's being distributed around the country. So we now have techniques to image T cells, you can do CD4 cells, you can image macrophages, you can image just about any type of cell population you want. And wouldn't it be amazing if we could see some of those results in the kinetics with early dynamic imaging, as opposed to just the static imaging, which we typically do. This is usually at 24 hours. We heard about drug development and the opportunities for that. The regulatory environment in Australia is incredibly well suited to performing phase 0 or phase 1 trials, to look at patient selection, drug dose selection, receptor occupancy, and so on. And I have to say that our own group in Melbourne, we can't do everything we're approached to do. We turn down probably 3/4 of the approaches that we have. This is an example of a bispecific which actually binds to PD-L1 and TGF-beta, TGF-beta being an immune suppressive molecule in the tumour microenvironment, where this company actually had a bispecific which could actually both activate by PD-L1 and reduce the immunosuppression. They wanted to know where it was going in the body. We did the animal biodistribution studies as well as the labelling to provide the proof to go into the clinic. And we actually showed that there was very little going into the tumour. This coincided with their phase 2 program being negative, and they cancelled. And the comment they made was if they'd done this study in their phase 1, that would have totally changed the way they would have developed that particular immunotherapy probe. So there are huge opportunities for you to be able to do this as part of the program that you have here. Very last thing I'd comment on is the opportunities through multi-center trials. ARPANET is one of the shining lights of molecular imaging multi-center studies around the world. The US, Europe, Asia, all want to copy the programs that we have here. And to be able to utilise the total body system here as part of this network, I think will be incredibly important. This program is run by Ros. Many of us have been involved with this for some time. And of course, we're working very closely with NIF to look at the imaging by repository approaches and opportunities to be able to analyse this. One of the first NIF studies that was ever done was led by Professor Roach. This is an incredibly highly cited paper looking at management impact of gallium PSMA PET in patients with prostate cancer. And this has led to a series of studies, including therapy studies, such as lutetium PSMA. The therapy study, along with the vision study, were the 2 international studies which have led to lutetium PSMA approval. The therapy study was an investigator-initiated study here in Australia. And we know that that allows us to be able to, from a context of trials, bring together multiple sites together. But I would encourage you to think also about some of the basic biology, the way you can upregulate targets, the way you can enhance radiation sensitivity, one of which was an example which was shown by Dale, as you can see here, where redifferentiation therapy in the context of radioiodine refractory thyroid cancer patients can markedly upregulate the uptake of radioiodine. And using I-124 with PET can provide the dosimetry which we otherwise can't easily do. This is such a beautiful image. I'll have to keep showing it if that's okay. And just as an example of the studies which are currently being done diagnostically within ARPANET and therapeutically within ARPANET, these are all trials which are investigator-initiated, funded primarily through MRFF or through philanthropic groups, and for which there's opportunities for this site to contribute enormously to some of the scientific data that we can generate through these studies. So this is my last slide. I think that translation of discoveries to the clinic is clearly one of the areas where total body PET system can play an enormous role. It's very important to have the linkage of the understanding of biology and physiology with the probes that can be utilised to answer these questions, and to take these forward through the network of radiochemistry facilities around the country, focused here to be able to explore further, and not just with a standard PET-CT scanner, but with your scanner here as well. So we look forward to seeing how the progress of the total body PET facility here progresses over the next few years. We're all very jealous. Hopefully we'll get a system in 5, 10 years from now. But in the meantime, we wish everyone the best, and if we can assist and collaborate in any way, that would be great. Thank you.